artificial selection. So I know that we kind of already talked about some of this, but we're going to review just because it was at the end of the class and I want to make sure we understand this. So what we've been talking about thus far is natural selection. And we talked about in all the things we've been doing, we have to have variation amongst the individuals. There has to be some differential survival so that some individuals are better suited to survive and reproduce. And when they reproduce, they pass on those variations that they had to the next generation. And so over a period of time, generation after generation, helpful traits, helpful variations tend to increase. Any variation that is harmful to the survival and reproduction decreases. And over a period of time, that leads to evolution, change in a species. And it can happen, oftentimes it takes a long period of time, but it can happen in shorter periods of time. And we saw that in the work that the Grants were doing on the Galapagos. They saw changes in the frequency of a thicker beak in just a couple years. Okay? And so if you imagine repeating that year after year, then evolution happens, species change over time. We also talked about speciation. When the species get split, often, there's other ways speciation happens, but often they get split geographically. Each group takes a different pathway in evolution, and eventually they become so different, they are no longer one species. Okay? And if they are introduced to each other again, they won't reproduce together. At that point, we say they become two separate species. So artificial selection is basically just like natural selection, but instead of nature and natural conditions choosing which organisms survive and which don't, it's humans deciding, humans choosing which individuals survive, which individuals breed, and through that process, people can actually change species over time. And most frequently talk about this in terms of like agriculture, okay? And domestication, domestic animals, farm animals, crops that we grow. All of those things have been artificially selected by humans for tens of thousands of years to have certain traits that we wanted, that we desired. And so artificial selective is sometimes called selective breeding because it's a person choosing which individuals are going to reproduce. All of the things we think about as like domestic animals and farm animals and crops that we eat, they all started as wild species that came about through natural selection. Humans found something about them that they liked, maybe an edible fruit that didn't make you sick, but then over time they took those and they used selective breeding, artificial selection to improve upon them in the view of humans. So this is an example. This is cauliflower, broccoli, cabbage, kale, kohlrabi. So these are all things I'm sure you're familiar with. They all derive from one wild ancestor. Wild mustard is the plant. It doesn't look like any of them really. It's pretty simple, small little plant, grows as a weed all over. But by selecting certain individuals to reproduce, humans have made all of these different vegetables. They're all derived from wild mustard. In cauliflower, it was bred so that the flowers are sterile and don't reproduce. And that produced these heads of cauliflower, sterile flowers. In broccoli, the flowers don't even develop. It was bred so that the flowers start to emerge and then stop development. And that's what those little circular things that broccoli is made of. Each one is a flower that didn't develop. It's been selectively bred so that it doesn't continue its development. In cabbage, the distance between the leaves, usually like in wild mustard, there's a big gap between any pair of leaves. Cabbage was bred so that that gap is reduced and reduced, and basically it's just a head of leaves, so that there's no real stem there. Okay. Kale is derived from mustard, which was bred to have larger and larger leaves. Kohlrabi was the lateral growing areas of the plant grow larger to produce that vegetable. So the idea is that humans did all these things. They created all of these vegetables from a single wild ancestor. Corn. Okay, corn comes from a native, originally a plant that's similar in size and shape. However, the fruits of this original I don't even know how to pronounce it, teosinte plant form these tiny little fruits, about the size of a quarter, that were probably edible in some way. You're not going to get a lot of nutrition from a tiny little quarter-sized fruit. But over many generations, humans took, the plants with the largest of those fruits and bred them together over and over again until we have what we call our modern corn today, which has huge fruits that have lots of sugar in them, that taste sweet. And so we bred those plants from wild ancestors. Dogs. Dogs are another great example. Dogs are domestic animals. They were selectively bred from wolves. And basically, if humans, probably killed, so there are wolves sort of around human settlements. Any aggressive animals, humans would kill because they could be dangerous. And possibly they allowed to sort of hang around sort of the most docile, the tamest of them. Maybe they fed them and allowed them to stay around camp. Eventually they breed together, generation after generation. You always choose the tamest, nicest little dogs to allow to survive. And over time you create domestic dogs. And then you take domestic dogs and humans chose certain traits, size, and they bred large dogs or small dogs. They took the smallest of these dogs, bred them together until you get smaller and smaller dogs. Dogs that could swim well, dogs that work, dogs that are protected. So basically by choosing the traits you like in these offspring and allowing them to mate, that's how you produce our modern dog breeds. All modern dog breeds, however, are one species. They're like a subspecies almost, a breed within that. They all can interbreed with each other, so they're not separate species. All right, another type of selection, type of natural selection, is something called sexual selection. This is when natural selection takes place, but it's favoring mating success. And so often, species that are evolved through sexual selection have evolved to have sexual dimorphism. And so that means that the males and females of these species have significant differences in appearance, or call, or behavior. And in most of these cases, the males have evolved to be more showy, more conspicuous, to have larger parts for decorative mating purposes. And females generally have evolved to be smaller, often more camouflage, more hidden, without all of these sort of showy characteristics. And so often it's males of a species that are more colorful, okay, have a larger tail. This is obviously just in birds, but the male wren, bright blue breast feathers, and then the female. If you ever seen like mallard ducks, like the ones with the green in their wing and the blue, they are the male ducks, and the female mallards just kind of look like brown spotted ducks. Peacocks. Peacocks are the males with huge tail feathers used to attract and mate with females. The females, the peahen, is just sort of a brownish color. Happens with lots of species, not just birds, fish, and woolly lizards with that showy dewlap from the males they use in their mating. Okay, lions, deer, antlers on male deer, no antlers on does. Okay, that's part of that sexual selection, and even insects as well. So that's sexual selection. So one of the things we often try to do as we're studying evolution is kind of trace back the history of a species and look at what were its ancestors, what organisms is it closely related to, how long has it been. since these two species had a common ancestor. Now those, we call that phylogeny, the tracing of the evolutionary history of a species. And that was often done based on just visual appearance, based on fossil evidence, morphology, the appearance, that's what means the appearance. But as we have improved upon our techniques of molecular biology, we now can use a much more certain techniques to determine how species are related. You know, I've used this example before, but if you were just to look at the appearance of a dolphin and a shark, you might say, well, they're probably closely related. They look very similar. They live in the water. They have fins, smooth bodies, all these characteristics, similar shape. But in actuality, they're not closely related. And so by comparing DNA or other molecular evidence, we can be much more sure of these relationships, and we also can be more precise. And so, do you guys remember, who did we say was the father of modern taxonomy? We said it in 7th grade. This guy that loved Latin. He changed his name. Carolus Linnaeus. So he was sort of the person that really started categorizing, putting things into these groups and through taxonomy. As modern molecular biology has advanced and we start looking at the DNA, we find lots of relationships that we thought existed no longer exist anymore. That we were wrong about the relationship between lots of species. So what we do to show this is we use what's called cladograms. Cladograms basically are like a family tree of species that shows how they're related. So in this cladogram, which is obviously very simple, what is it telling us? What does this? It tells us several things about a human, a mushroom, and a plant. Grace?
Humans and mushrooms share a common ancestor.
Correct. Where was their common ancestor? Like on this cladogram? Right there. Right? This is when, if we trace back the line for the human and fungus, they had a common ancestor. Okay, give me another piece of information we can gather from this cladogram. Does it tell you anything else? Right?
Human and mushrooms are fungus are more related than plants and fungus.
Yes, that humans, fungus is more closely related to humans than it is to plants, and that's true. That's something that scientists, again, didn't think until we started analyzing molecular evidence. They would have said that the mushroom is more closely related to a plant, but actually it's not. Did plants and mushrooms ever share a common ancestor? Yes, they did, here. Okay. So that's how these diagrams work. They obviously can be much more complex than this. Okay. So again, these are mammals in the order Carnivora. Which includes all of these animals. They're all in that same order. Okay. And so if you look at them, we see they all did derive from a common ancestor at some point in the past. As this common ancestor branched off, it formed the cats, and the Stellidae family of like otters and fishers and wolverines, skunks, otters, something people, ferrets, that's in this group. And then the family of dogs, they split off. So we know dogs and wolves are related. What's closely related? A skunk and the dog, or the skunk and the panther? What's the skunk more closely related to? Grady? The dog. Because if you trace back the skunk, had a common ancestor with the dog here. You have to go back a little bit farther to find the common ancestor of the leopard. Okay, and so for our last topic to talk about, let's talk about life on Earth. So we know that life on Earth has evolved into the diversity of species we have today. We know that lots of species have become extinct over time, new species arise, and so forth. So this is sort of scientists' current best hypothesis about the evolution of life on Earth. It's not, so we're more sure of the later stages and less sure of the earlier stages. So we think about the initial, so we know the early Earth, when the Earth had just formed, was quite different than it was today. So the question scientists were asking was, well, where did the first organic molecules, like DNA and proteins and carbohydrates, where did they come from? Okay? We know that today, organic molecules do not just come about from inorganic molecules. And you need organic molecules for life. So where did they come from? Very famous experiment was done by two scientists, Miller and Urey, in the 1950s. And what they did is they tried to simulate what scientists knew about the early Earth's atmosphere. So the early Earth's atmosphere was much different. There was no oxygen. There was immense amounts of energy and lightning. There were different chemicals in the water and in the atmosphere. So what they did is they took a sterile environment that they thought was similar to the Earth's early atmosphere. That's what's here. This mixture of various gases. Okay. They recreated what they thought the oceans were like in the Earth's beginning. Okay, they kept it all contained, where they were nothing else was added. So they basically just had water, okay, some simple gases, okay, and then they added electrical charge to it, sparks, which were meant to simulate lighting. And they did this, kept it on these conditions. What they did is they found, after this experiment, in the liquid, they found that these simple molecules that they had started with had formed some more complex organic molecules. Things like amino acids, things you must have for life. And so they recreated an experiment that you can get organic compounds from inorganic compounds, such as were probably present on the early Earth. And so the thinking was that we had basic, simple inorganic compounds, which combined with the energy that was present to form more complex compounds, which combined to form things like nucleotides, eventually DNA and RNA. That's the thoughts about how that happened. The first known life on Earth was bacteria-like organisms. And this is 3.5 billion years ago. So about 1 billion years after the Earth was formed. So how would these first cells arise? So first off, some things to know, that there was no oxygen on the Earth. Okay, so scientists believe that the first types of living things used anaerobic respiration. Cellular respiration does not require oxygen. It's similar to fermentation, what they call it today. This is called the heterotroph hypothesis. The first life on Earth were heterotrophs. And eventually, these heterotrophic organisms, much like bacteria, evolved after, about 0.7 billion years into photosynthetic bacteria. Bacteria that could create their own complex molecules for energy. Once that shift happened, we know photosynthesis releases oxygen. Oxygen began to accumulate in the atmosphere. And eventually that led to organisms which could use that oxygen in respiration. And that's when the evolution of aerobic respiration happened. In aerobic respiration, using oxygen, you get much more energy for molecules than you can in anaerobic respiration. So that brings us to like bacteria, basically. At this point, we have just bacteria. So what do we call bacteria and other cells without organelles, without a nucleus? There's a name for them. No, prokaryotic. They are prokaryotic cells. Bacteria today are still prokaryotic. They have no organelles within them. They have no nucleus. So the question scientists had to ask was, well, then how do we have these more complex cells today? How would they have evolved? And the theory they came up with is this endosymbiotic theory, which said that basically we had billions of years ago, we had basic prokaryotes, simple cells that had a membrane, some cytoplasm, and some DNA. Things like bacteria. Over time, this outer membrane kind of started folding inside. And that's what led to the formation of the nucleus that surrounded the DNA. Then, scientists think that in some way, a heterotrophic bacteria evolved to have a symbiotic relationship with one of these cells. And this basically is a bacteria cell, started living inside of this cell. And what do you think it became? This aerobic, heterotrophic bacteria that lives inside a cell. And guess what it eventually evolved into? Grace? Mitochondria. Mitochondria use oxygen to get energy. This became mitochondria. Also, an autotrophic bacteria evolved a similar sort of situation, entered into these cells, and began living there. That photosynthetic bacteria became what? What organelle would that be in a cell that carries out photosynthesis? Come on. That became chloroplast, yes. And so what we have is this, now we would call it a eukaryotic cell with organelles, with a nucleus, evolved by prokaryotic cells taking in these small bacteria and developing this endosymbiotic relationship with them. From there, we have simple single-cell eukaryotic organisms. Eventually, sexual reproduction formed, in which you had cells exchanging this genetic material. Organisms evolved to be made of more than one cell. That's multicellularity. Multicellular organisms evolved into simple invertebrates, like sponges, worms, jellyfish. Eventually, plants evolved from those photosynthetic eukaryotes. Vertebrates evolved, first jawless fish, then jawed fish. Some of those fish evolved to be able to live life on land as amphibians, which evolved into animals that live permanently on land. Reptiles, which split and evolved into both birds and mammals along the way of plants as well. So this shows sort of the more recent stages in evolution of life on Earth. So this goes back about 650 million years ago. The first animals back in the beginning of those stages. Soft body animals. Then you have the development of vertebrates, chordates, fish. You have plants moving onto land. You have fish evolving to jawed fish, and the first amphibians developing. Them, the reptiles developed, trees, ferns. Reptiles diversified in the Permian period into a wide range. The first dinosaurs came about in the Triassic period. They diversified and sort of dominated the Earth's land masses in the Jurassic period. That's the name, Jurassic Park. First birds evolved at the end of that period. As dinosaurs became extinct, that opened up for the evolution and diversification of mammals. Eventually, the before the evolution of humans happened, much more recent past several million years. So that's sort of the short and sweet evolution of life on Earth. Any questions about any of our notes on evolution or natural selection? Okay. So we're done with our notes. We still have 3 more labs to do in this unit. And then we'll have a test. Probably look at a test, not next week, I would guess the week after that. I'm going to prepare a review packet for you, and then we'll be moving on. So with the remaining time today, you can work on your bench if you'd like.